Okay, it's now time for the last, but certainly not the least. I'd like to welcome Dr. Anna Grenha to the podium. Anna is, of course, a very valuable member of the DDL Organising Committee. A quick introduction. Anna received a PhD in Pharmacy and Pharmaceutical Technology from the University of Santiago de Compostela, I hope that's right, in 2007. She's an assistant professor in pharmaceutical technology at the University of Algarve in Portugal since 2007. She is the PI in the Drug Delivery Laboratory, which is dedicated to the design and the development of particulate carriers for drug delivery, with particular focus on inhalation. And the research group is keen in exploring the potential of natural materials, namely polysaccharides, fostering new applications that benefit from their intrinsic properties. So with that, over to you, Anna.
Thank you, Gary. Thank you very much for the nice introduction. And good morning to everyone. It is my great pleasure to be here. And I would like to start by thanking the opportunity that was given to me to be here today and to share with you some of the research we have been conducting in my research group at the University of Algarve. I also know that it is almost lunchtime, so you are all willing to go. And I got this great responsibility and the honor to close the works of this conference, but on top of that, my voice is kind of fading away, so I hope it subsists until the end of the time, and I promise I will be quick, and hope I am up to the task. So I'm going to talk about polysaccharides, and in the end of my talk, I hope I have guided you to the answer to the question that I pose in the title of my communication, of if there is any opportunity for the application of polysaccharides as lung delivery excipients. So we all know, and it has been said repeatedly today, that the list of excipients that are approved for lung delivery is quite short. And it hasn't been changing much in the last years, has it? In fact, not. And this happens because several reasons, either because the requirements for the approval of new materials are not perhaps very clear, as we saw today earlier in the morning, or even because naturally the companies just realize that including new excipients in their formulations will naturally lead to more extended processes of approval and also to an increase in the cost of the final formulations. But on the contrary, if we look at the literature, it is quite rich in applications that involve new materials. And this is somehow a contradiction, isn't it? I think that the fact that the literature is quite rich in this, the involvement of new materials is because we are nowadays focusing a lot more on dry powder formulations and also on the use of new technologies like particle engineering and others that we have seen today here in the morning. So these kind of situations are perhaps more amenable for the inclusion of new materials, and so they are just coming up. But it is a fact that there is a shortage of approved materials. This shortage is certainly impairing the progress of our field. And because of that, there is a pressing need for the inclusion of new excipients in the formulations, because we all know that, at least partially, that is the way, including new materials, is the way we have to have differentiating approaches, to have different formulations, and to take benefit from the functional abilities that we can identify in our materials. And ultimately, this will lead certainly to improved therapies that we can design and develop. So the literature describes rather different materials, from lipids to proteins to synthetic polymers and also to polysaccharides. And this is quite a popular class of materials nowadays. Polysaccharides correspond to complex carbohydrates composed of monomers, of monosaccharides like glucose or galactose that just join together to end up in a final larger molecule that comprises the polysaccharide. And they are natural materials, and they can be obtained from different sources and varied sources, like from plants, from animals, from microbes, inclusive from algae, and they have quite appealing properties. They are structurally flexible, so kind of easy to work with. They can be easily functionalized. They are potentially biodegradable, and in some cases even endogenous to the human organism, and they can be obtained at a relatively low cost. But nevertheless, they also have some limitations that we need to take into account when we select this class of materials for any application. And I would say that the greatest one of these limitations corresponds to the batch-to-batch variation, which happens because there are different culturing conditions, for instance, or because we use different extraction conditions to obtain the polysaccharides from the source materials. On being natural materials, they are also more prone to microbial contamination, and there is associated a toxicological and an immunological risk on their use and their application. So polysaccharides are described to have a lot of abilities, from the most reported ability to control the release, as in this example here, where there is a controlled release of a model cytotoxic drug, 5-fluorouracil, from chondroitin sulfate microparticles compared to the dissolution profile of the free drug, to other abilities like the one exemplified in this graphic, where we can see, let me see if I can use this, yes, that the retention time of salbutamol sulfate is quite increased in the lung tissue when we deliver this salbutamol sulfate microencapsulated in hyaluronic acid microparticles as compared to the free drug, which has a much lower retention time. And this is also beneficial for the lower systemic exposure of the drug, which is desired in this case. So this effect was attributed, in fact, to the mucoadhesive properties of hyaluronic acid microparticles. So in our group, we have also been working a lot with polysaccharides, some of them very popular, like chitosan, but many others that are quite unknown, or at least less known materials, and I described some of them here: locust bean gum, fucoidan, glucomannan from konjac, and chondroitin sulfate are some of the polymers we have been working with. So I will showcase the abilities and the positive properties of these, some of these polymers, using the example of an inhalable tuberculosis therapy that we developed. Tuberculosis is a respiratory infection that occurs when an healthy individual just inhales droplets coming from an infected individual that are contaminated with the infecting bacteria. And after the inhalation of these droplets, if the common mechanisms of defense of the lung cannot eliminate the bacteria, they will just go through the lung and will arrive to the alveolar zone, where they will be mostly phagocytosed by the alveolar macrophages. And once inside, they will find a way to replicate and to be hosted by these cells. So what we thought is that, considering, and this is widely described in the literature, wasn't an original idea from our group, but if it is a local disease of the lung, it should be kind of logical to try to provide the direct delivery of the drugs to the lung instead of using the conventional oral therapy. So we just developed a formulation approach where we microencapsulated anti-tubercular drugs that are currently in clinics in order to make them reach the alveolar zone. So the particles that were produced were adapted to show the aerodynamics suitable for to reach the target zone, the alveolar zone. But on top of that, we wanted to go one step further, and we wanted to carefully select the materials we were using as matrix of the microparticles in order to provide a favorable or a more favorable interaction between the microparticles and the macrophages, which are the hosts of the infecting bacteria. So the literature describes that these macrophages have specific surface receptors, including C-type lectin receptors like mannose receptor and also scavenger receptors. So we worked with polysaccharides that, at least theoretically, had a higher probability of having a close contact with the macrophages mediated by this interaction with the surface receptors. So I will show you the results that we obtained for two different polysaccharides, glucomannan from konjac and locust bean gum. Locust bean gum is a galactomannan and is a regional product of the Algarve. We produced the microparticles with each of these polysaccharides co-associating to antitubercular drugs that are currently in clinics, isoniazid and rifabutin, and we produced the microparticles by spray drying. This is the morphological aspect of the microparticles, quite spherical, but with very high convexity. So, in fact, for glucomannan microparticles, we have characterized this convexity parameter, and it is quite close to 1, which is the maximum. So, theoretically, at least, it is beneficial for aerodynamic properties. And we have managed to associate the drugs efficiently to the microparticles, and regarding the aerodynamic parameters, we had emitted doses above 90% and fine particle fractions that are roughly between 40 and 55%. So one of the most important assays for us to perform was a validation of our theory, not only for this application in tuberculosis, because we were running also others, so to validate the ability that our materials could have to mediate a good contact, a favorable contact with the macrophages. So we performed a flow cytometry assay. We used monocytes, human monocytes, THP1 cells that we differentiated to acquire the macrophage phenotype, and then we stained both the cells and the microparticles with different fluorescent labels, and we exposed the cells to the dry powders for a period of 2 hours. And when we did the flow cytometry assay, we realized that both polymers were providing a percentage of cells evidencing signs of phagocytic activity around 90–100%. And we were quite satisfied about that. These are the results of other materials we were testing, just to show you that with chondroitin sulfate, for instance, and fucoidan, we obtained quite lower results, while all guagam and chitosan also performed very well regarding this interaction. We performed an in vivo proof of concept with these microparticles in a murine model of the tuberculosis infection, and after observing that the infection was established, we initiated a period of treatment of 2 weeks using our microparticles of locust bean gum. So we established 4 groups, 1 untreated, 1 receiving the co-association of drugs by oral delivery 3 times per week, 10 milligrams per kilo of each drug, and 2 groups on inhalation, 1 receiving 3 times per week the inhalation, and another one 5 times per week at lower doses of the drugs. We determined the growth index, and we were very pleased to see that the inhalation 5 times per week was the only therapeutic schedule that allowed us to have a negative growth index in the 3 organs that were being analyzed, the lung, the spleen, and liver, and above all, in the lung, because it was our target organ. Just to clarify, the growth index and obtaining a negative growth index means that we decrease the number of bacteria comparing with the beginning of the treatment. The histological analysis also supports these observations, as we can see in the group receiving 5 times per week the microparticles. We see no microscopic lesions in the lung, while both spleen and liver are still evidencing signs of some macrophage groups or even granulomas. When we got to this point, we just realized that we had quite interesting results, but in fact, we were missing a lot of information on the toxicity of the materials. And this links perfectly with our first talk in this section from Victoria MacDonald. Because we really need to characterize the toxicity. It's a very important parameter for these new materials, and for all, but especially for these, which have no information regarding toxicity. So we started with a very basic approach of in vitro cell viability, but we faced a limitation because we wanted to have biorelevant assay conditions. That means to deliver the powders directly to the cell, to the cell culture, and we had no device that allowed us to aerosolize the powder over the cells. I know there are some in the market that could be adapted for the effect, but we couldn't afford it. So it was not a possibility, and we just decided to develop our own device, which allowed us then to culture the cells in a Petri dish, and the device perfectly fits the Petri dish, and with the help of an air compressor, we can just aerosolize the dry powders over the cell culture. So using this approach, we tested the cell viability of locust bean gum microparticles. We used A549 cells and the alveolar epithelial cell lines as a model of respiratory cells, and we tested the locust bean gum microparticles at a dose of 125 micrograms per square centimeter, both at 3 hours and 24 hours, and we could see that even after 24 hours of exposure, we are still around 80% of cell viability. We proceeded with an in vivo assessment of these microparticles. I want to reinforce that these microparticles are locust bean gum microparticles only, so there is no drug. We are just trying to process the microparticles, to process the material as we use for applications, but testing only the effect of the material. So we used bulb C mice, and we exposed the animals to an inhalation of these microparticles at a dose of 240 milligrams per kilo. We established 3 groups, one naive, one receiving only 1 dose, which is the acute exposure, and another one receiving 10 doses of the microparticles for a period of 2 weeks. We also performed a similar study for konjac glucomannan, but the results were quite similar. I will just show you the results of locust bean gum. So the evolution of the body weight of the animals showed no signs of toxicity, no differences were obtained. But when we go to the leukocyte count in blood, we can see that the number of lymphocytes and the number of neutrophils are actually evidencing a statistically significant difference after the inhalation of 10 doses. And although not statistically significant, eosinophils also increased a little bit after the inhalation of the 10 doses. So this can be an indication of an inflammatory or an allergic response, and we really want to go deeper on this analysis to try to obtain more information. We also tested and quantified the IgE in plasma, but there was no significant difference, as you can see. The same happened for the tissue index of the 4 organs that we were analyzing: liver, spleen, lung, and kidney, and the same for histology. So the histological analysis, when we compare the naive animals with the animals receiving 10 doses of locust bean gum microparticles, didn't show any signs of alterations. So with this, I'm approaching the end of the talk, and I hope that your answer to the question that I posed at the beginning is that definitely we have a potential, not only regarding this targeting ability that I was showing, but there are many other abilities that polysaccharides have and that we should explore. And I would like to leave you with a take-home message. I know that there are a lot of people present that are formulators that work in formulation. So my message is that we need to work hard to keep producing evidence, to discuss this evidence with pharma companies, and perhaps with that we will do the effort altogether to have more excipients in the market. So I would like to thank to my team and to the collaborations that enabled this research, and also to the funding bodies. And that's it. Thank you very much. My voice persisted. I'm very glad, and I'm available to answer your questions. Thank you very much.
Thank you, Anna. Very comprehensive. Great. Okay, folks, any questions for Anna? Well, one on Slido. Just wait and see if we have any live people. Okay, we'll take this one first. Can you comment on I can't even pronounce that. That's the one. Issues are using polysaccharides, mostly natural, for inhalation.
Can I comment on polysaccharides natural? I didn't understand.
Yeah, have you got any comments on the immunogenicity of polysaccharides by the inhaled route?
I promise I didn't understand. The what?
Immunogenicity?
The immunogenicity. Oh, now.
That's my accent,
Sorry.
sorry.
Yes, we all know that these kind of substances actually have some immunogenic potential, so we need to treat this very carefully. And this is actually one of the reasons that after we developed the initial project of tuberculosis, and as you know, we showed some in vivo studies with 2 weeks of treatment, but that is definitely very short. It was all we could perform and afford, but we certainly, even if we are using inhalation, we will have more extended treatments, and that's one of the parameters I started to think about a lot, the immunogenic potential that persistent doses of these materials will have on the lung. Actually, I think that's a very important parameter that we need to take into consideration.
Okay, thank you very much, Anna.